Our next speaker, Karen Dannemiller, I may have butchered your name, from Ohio State University. I even heard your name this morning, so I should have gotten it right.
Hello, everyone. Let me see. Okay. So if we get sick, we go to the doctor and we probably get some medication. And the same is true for the 8% of the US population who has asthma. But I want to ask the question, is this always the best way to go? Especially for children who have asthma, a lot of these medications can have potentially harmful side effects, and it's still controversial, but they may even permanently stunt the growth of these children. But we have an opportunity here that I think is fairly underutilized to also control the environment of these children and think about the triggers that they're exposed to, which may help reduce some of the medications that they're on. And that's a common goal of a lot of these children. And one important trigger that a lot of these children respond to that we need to understand more about is mold and housing. So exposure of asthmatics to mold and housing alone costs an estimated $3.5 billion in the United States every year, and it's usually not as pretty as this piece of artwork over here. A lot of our exposure to these microbes comes from dust, as you've heard about in a lot of other talks, and so that's one area that I'm going to focus on, where we have about 83% of airborne bacteria coming from indoor resuspension of dust from the floor, and about 66% of airborne fungi. So my talk is really going to focus on trying to put a lot of these pieces together, and the first part is focused on linking these indoor microbial communities to human health effects, specifically childhood asthma, and then the second part is looking at how these housing characteristics can then influence microbial communities in ways that may be important to health, and there I'm going to focus on moisture in housing. So there are a lot of studies looking at some of these microbial exposures in asthma. You heard about some of them earlier, and I'm going to highlight two fairly quickly today. The first is the STAR study that we worked on in Connecticut and Massachusetts, where we enrolled 200 kids with asthma, followed them over the course of a month to look at their asthma severity, and then measured the fungi and bacteria that were present in their dust samples. And a little bit of background information. Asthma has multiple different phenotypes, but broadly you could characterize it as allergic asthma or non-allergic asthma. And these are mediated by different mechanisms in the lungs. So allergic asthma involves eosinophils and involves IgE and is sort of an allergic type response, where non-allergic asthma is mediated by neutrophils and is an acute inflammatory response. You could think of it as more of an irritant type response. And so in our study, we divided the kids into these two groups, allergic and non-allergic asthma, and looked at their microbial exposures. And what we found is that it was important what type of asthma the child had and how they were responding to these microbial communities. So both did respond to the microbial communities, but for the allergic children, it mattered more what they were exposed to. So if they were exposed to a lot of allergenic fungal species, but it didn't necessarily matter how much. So you can see that over here. We didn't see an association there. For the non-allergic kids, it didn't matter what they were exposed to necessarily. We didn't find an association over there, but it mattered how much. So think of those kids as having more of an irritant type response, which makes a lot of sense with this data. So for the allergic kids, it mattered what they were exposed to, and for the non-allergic kids, it mattered how much. So we see both responding to these indoor microbial communities, but responding in different ways. I'm also working with collaborators at Columbia University, including Matthew Perzanowski, on looking at the New York City neighborhood asthma and allergy study. And this is looking at 500 dust samples collected from homes of children, and we have health information and housing characteristics. And this was an interesting study because the asthma prevalence rates are really different in different neighborhoods in New York City. So you can see on this graph they range from about 2.6% in some of the beige areas up to about 18% in some of the dark orange areas. And so we looked at the differences in microbial communities that were present in these neighborhoods. And this data is fairly hot off the presses, so we're still analyzing for health information, but our preliminary data shows an association with low fungal diversity on an ecological level in these neighborhoods being associated with increased asthma prevalence in these neighborhoods. This is still sort of an interesting finding that's been found in other studies. I think Mark Mandel is going to talk a little bit more about that after me. And we see a similar association with asthma persistence. So we're actively analyzing this data. Here's a spoiler of things to come in the future. And we're also continuing the taxonomic analysis, looking specifically at things like Cryptococcus, which we also found in our previous study. So from this work, I've just highlighted just these two studies. We saw associations between the indoor microbiome and asthma severity in children. And this can be taken into context, like I said, the multiple other studies out there, to try to identify what we need to look for in these houses and try to figure out what we can do about it in terms of reducing environmental exposures to known asthma triggers. So now I'm going to move on to part 2, looking at these microbes in homes and how they're related to both moisture and how this could potentially influence health of children that are exposed. So here's some lovely pictures for you of moldy building materials. So in the same study done in Connecticut and Massachusetts, we looked at housing characteristics, and I'm not going to go through the details here. Others have alluded to this earlier, but we found that major influences were ventilation, occupancy, pets, and moisture on the indoor microbial communities that were found in these buildings. So this indicates that we need to think about how we're designing our buildings and how that might be influencing these indoor microbial communities and our exposure. In the New York City study, we have some additional data on things like carpet, and we see a pretty dramatic influence on the fungal communities based on whether a home had carpet in it or not. In fact, we found 205 fungal species that were identified that were associated with having a carpet in your home, and only 4 that were associated with less. And you can see a list of them up here. I won't go through this whole list, but some of these are species that potentially contain allergens. So when we put something like a carpet in a home, we're changing what our exposure looks like, not only through increased resuspension from these carpets, but also what's actually present. Building type also had an influence, whether you were in a single family, a multifamily unit, or a larger apartment building. And then we also looked at flooding. So flooding was also really important. We saw differences in homes that were flooded from Hurricane Sandy in New York City. 56 species were identified as more prevalent in these flooded homes. You can see a list of just a subset of them up there. And they're things that you would expect to find in a moisture damaged building, things like Aspergillus and Penicillium. Nothing too surprising there. But so this begs the question, we want to ask a little bit more about moisture and fundamentally what's happening in our buildings when we have situations of increased moisture. So we conducted a study where we got used carpet from homes. This is an example of one of the carpets that we got. This is a 10-year-old nylon carpet that we pulled out of a house, got dust from that same house, embedded it in the carpet, and exposed it to increased relative humidity conditions because we wanted to better understand what this moisture was doing in this house and how this might be influencing microbial communities, ideally to then be able to link that back to health outcomes. And we weren't sure, actually, starting this, if anything was going to grow at all. But indeed it did. So on this graph, you have the concentration of fungi in blue and bacteria in red on the y-axis, and the relative humidity that we incubated these samples at on the x-axis. And you can see that at increased relative humidity, you get increased concentration of fungi starting at around 80%, and bacteria we only saw that increase in concentration at 100%. This was for a 1-week incubation, and I think we may have even seen increased growth at lower relative humidities if we had waited a little bit longer. So those experiments are underway. We continued these experiments. The goal was to continue these experiments out for 6 weeks at both 100% and 85% relative humidity. But the carpet coupons incubated at 100% relative humidity were so entirely covered in visible fungal growth at 3 weeks that we decided that was enough. We had shown that it was growing there. But you can see that this growth continues out up to about 6 weeks. So now the question is, these are fairly high relative humidity levels that we had looked at in these homes, but they're also not unreasonable. They might be something you might find in a hot, humid climate with no air conditioning, or in a home after an event such as showering, where relative humidity might increase. But nobody's home is going to sit exactly at 85% relative humidity for a week at a time. So we wanted to look at diurnal variations that naturally occur in these homes. So we conducted a second study where we varied relative humidity. An example graph is up here. You have relative humidity on the y-axis and the number of days on the x-axis, and we kept it at elevated relative humidity levels for specific periods of time. And we saw in this carpet, as we did in the previous carpet, you get a lot of fungal species showing up that are expected in damp buildings, so things like aspergillus and penicillium again, that are showing up with increased moisture for increased periods of time. And so our question is, this is great. These things still grow even with diurnal variations that naturally occur in buildings, but can we actually model this? Coming from an engineering standpoint, can we put a mathematical model to this? So we looked at a mathematical model that was developed by Olaf Aden for growth on drywall. And in this model, we have time of wetness on the x-axis, and this is basically think of it as the percentage of time that the material spends above some critical growth threshold. And then we have our relative growth rate over here on the y-axis. And our standard model would say the longer time period that you spend above this critical moisture growth threshold, the more growth you're going to get, the higher relative growth rate. Oops. Where did my graph go? Is there another graph on there? It's a really pretty graph. So anyways, we're pretty proud of this graph. It fits very nicely. I'll just explain what it looks like, and you could see it actually fits really nicely along this standard model. That we can actually model this fungal growth in the indoor environment and show that at higher increased time of wetness, we do get increased relative growth rates, and this is something that we can actually calculate out. And this is something that could be valuable for a lot of future studies to understand how moisture and housing is going to influence our indoor microbial exposures. So next we wanted to ask the question, okay, we've shown these things grow in the indoor environment, especially under these elevated relative humidity conditions, but what about microbial function? What are these things doing? So we did a metatranscriptomic analysis on this incubated dust. So we incubated this dust at 50%, 85%, and 100% relative humidity in these chambers, extracted the RNA, and saw what we got. And on this graph on the left, you can see similar clusters are going to cluster, similar samples are going to cluster together. And first of all, we were surprised. We actually got RNA out of dust that was incubated for a week at 50% relative humidity, which surprised us. We thought that we might not get anything, but indeed you do, and it's fairly high. It looks like high quality RNA where we have, lipid metabolism is complete, carbohydrate metabolism is complete. But we do see differences in our samples that were incubated at 50% versus 100% relative humidity. And the samples over here at 50% and 85% cluster together. They look fairly similar. And the samples incubated at 100% are not only very different from the samples at the lower moisture levels, but they're also very different from each other, which we thought was really interesting. So you sort of get this, I guess I'll call it metabolic diversity that's happening here, where they look different. And this is seen on the heat map on the right, where different samples that are more different are in blue. And you can see the samples at 100% look different, not only from those incubated at 50% and 85%, but they also look very different from each other. And this was the exact same carpet with the exact same dust, all started at the same time. So that was kind of surprising. Oh good, this works. So this is now my favorite GIF. I'm pretty sure it's going to go viral on Facebook. You'll see it soon. But what you can see is up here on the left is our incubation levels, and this is the KEGG ontology map that we have. And at 50% relative humidity incubation, we see mostly primary metabolic pathways that are lighting up. So things like lipid metabolism and carbohydrate metabolism we see are complete. At 85%, we see a little bit more, but still mostly these indoor microbes are focused on primary metabolic pathways. And then at 100%, we see that these microbes are transcribing a lot more of their genome. They're actually involved in a lot of secondary metabolic pathways. And which makes a lot of sense. So when they're moisture limited, they're really focused on those key functions that they really need to be doing, and then they expand out as more moisture becomes available. So we asked, how does this matter for human health? Does this have implications? Does this mean more allergens or more toxins are being produced? So we looked at contigs associated with allergens in our data set. And this is a violin plot on the left where you can see fold change either upregulated or downregulated, comparing 100% to 50%, 100% to 85, and 85% to 50. And indeed, we actually do see 2 times as much upregulation of contigs associated with allergens at 100% relative humidity compared to either 50 or 85. And so indeed it does seem like this fungal community is transcribing more genes associated with known allergens. So this is all RNA. We wanted to look at protein as well and say, do we see the same thing? So we ran an assay over here looking at allergenicity per spore on the y-axis. And we showed that those fungal species, this was a pure culture analysis, those incubated at 100% or the higher water activity levels produced actually more allergens per spore when they were in an ELISA against human IgE. So we actually saw an increase in this as well of the actual allergens that were produced in this species. So this begs the question, is a spore a spore? We're looking at all of these species in these samples, but it might also really depend on how these things are grown and where this actually came from. And you could think about what this means, going back to these kids with asthma. So we know that these kids with asthma, there's going to be different effects whether they have things like allergic or non-allergic asthma. You could hypothesize something like the kids with allergic asthma, it's going to matter that there's more allergens present. Maybe the kids with non-allergic asthma, it matters that there's more fungi grown in these moist conditions. But this needs to be an active area of research. It's something that we're excited to look into. So in summary, we saw an association between the indoor microbiome and human health. We also saw that home design choices can influence our indoor microbiome, and this is especially true with moisture, where this can play a critical role in both what we're exposed to and also microbial function indoors. So this is kind of bringing it all together. I have a lot of people to acknowledge for this work, especially the Alfred P. Sloan Foundation, who funded a lot of this work, and all of my collaborators and students who have been great to work with. So thank you.
I think we're going to have to skip questions. That was a very full talk. Next meeting, they need to give you a plenary or a keynote.